Welcome to the lecture series on regulatory science. I'm Christian Schneider. I'm Head of Biopharma Excellence and Chief Medical Officer for Biopharma at PharmaLex, which is a consultancy company. I'm an ex-regulator. Maybe just very briefly about me. I'm located in the north of Denmark in a city called Aalborg. So it's even further north than Hanover is. And just very briefly, I'm not going through this. I was at the Medicines and Healthcare Products Regulatory Agency, the MHRA, for a number of years in the UK. I was at the Danish Medicines Agency, but I'm originally German, as you can also hear from my accent. So I worked for the Paul-Ehrlich Institute, which is the German regulatory agency for biological medicines. And I had a number of roles in the European Medicines Agency as well. And this lecture series, as you might have seen from the program, has a number of different topics, more or less about early translation of non-clinical findings of research into the clinic, so clinical trials, for example, but then also further on in development of medicines. As we have seen, there are many researchers, obviously, in Hanover. One of the hearts of Hanover is research. So we found it a good opportunity to also talk about regulation and regulatory science at Hanover Medical School and the Twin Core. So that is the background for this lecture series. And I'll talk about risk-based approaches. A risk-based approach is almost a buzzword, and it's many people are using the term risk. Regulators used to use the term risk to make something that is risky look even more risky sometimes. But I will tell you today that this is not necessarily the case. So let's talk about the concept of risk-based approaches. An example for effective risk mitigation, and also the risk-based approach for advanced therapies. Advanced therapies is gene therapy, cell therapy, and tissue engineering according to the European law. And there we have even a guideline on risk-based approaches. Now, I'm sure all of you know Harry Potter, and I have this here because Harry Potter is, I think, one of the few in the movie and in the book, obviously, who dares to say the name Voldemort. Others are always saying that the person whose name should not be mentioned. And when he's asked once why he does that, he says, well, it's sometimes better to name something than it looks less scary. And it's the same with risk. If you name it, and it's not just identifying risks and that's it, and we have to stop development of a medicine that is maybe otherwise promising. No, this goes further into risk assessment. I'll talk about a bit more about this, about risk mitigation, so we can meet that risk and hopefully mitigate it. And then the assessment of effectiveness of that risk mitigation if necessary. And then if there is new risks coming up, we can then re-identify the risks, reassess the risks, and so on. It's a bit of a circle, you could say. And an example for a risk-based approach is unwanted immunogenicity. So for biological medicines like monoclonal antibodies or therapeutic proteins like erythropoietin, when there's always a possibility that the immune system, even if it is a fully human protein, recognizes something on the molecule for sometimes unknown, sometimes known reasons, and kind of attacks it and neutralizes it. And that has led to one of the paradigm cases more than 20 years ago, where with an erythropoietin, pure red cell aplasia was described in patients. So basically what happens here is there was an immune response against erythropoietin, which cross-reacted with the body's own residual erythropoietin that was left, and by this it's completely neutralized, so the bone marrow system doesn't get any stimulus anymore and just stops producing. And by this you have an aplasia, so an absence of cells of only the red cell lineage, because its erythropoietin is only working on the red cell lineage. So it's a pure red cell aplasia. And that, of course, had a very, very severe anemia in the patients They needed to have blood transfusions on a regular basis. So that was a little bit of a shock moment, you could say. And this is why unwanted immunogenicity has always been mentioned as a risk for biological medicines. But the question is it really always a major concern? Now, what is a risk? A risk is defined as the probability multiplied by the consequences. What does that mean? So we identify the risk first. In unwanted immunogenicity, there's a number of product-related factors and a number of patient-related factors which could lead to a higher predisposition on unwanted immunogenicity. So aggregates, for example, that was with erythropoietins, typically aggregates, which then form even either neoepitopes, or there are some sort of an adjuvanted presentation to the immune system. The structure itself, if it has murine sequences, for example, the expression system, and a number of other product-related factors can obviously trigger an immune response. And also patient-related factors that could be intrinsic factors like the underlying disease that's treated, extrinsic factors like previous or concomitant medication, the route of administration. So, for example, subcutaneous administration is sometimes mentioned as being more risky as intravenous administration, previous exposure, so there's some presensitization, and of course many other factors, which some of them we might not even know yet. So that impacts the probability of an immune response, and the consequences is there, for example, an endogenous counterpart, like with erythropoietin, a monoclonal antibody for therapy of a disease does not necessarily have an endogenous counterpart, because the antibodies are more or less against the antigen recognizing region of the antibody, and that is unique. It's not against the constant part, the Fc part, which is common to all immunoglobulins. But also, is there a therapeutic alternative that patients can use? How vulnerable are the patients? There's a number of different factors again, and you see that this is already going into the risk assessment as well. What does it mean for the particular patient? And this is why there is also the possibility to then predict the risk with certain methods. This is a known industry, you could say, that has been established following these concerns. And there's always available knowledge as well. There's an accumulation of data. So that risk-based approach is always a living concept. And once a risk has been determined, it could very well be that there is no higher risk anymore at some point, and then it's not a risk, and a risk-based approach is not necessary anymore, and it's mitigated. Now, as I said, the risk assessment is then the translation of findings. What does it mean for the patients? Is it a chronic disease, life-threatening disease? So is there any alternative at all? If there's no alternative, if it's a life-threatening disease, we might not even have an alternative then to treat the patients. But of course, within technical capability and feasibility. So regulators, when they ask about risk assessment, sometimes it has to be feasible what companies are asked for. So for example, to characterize the entire immune response against the therapeutic protein, including subtypes IgM, IgG, IgE, IgA, and so on, might be a bit over the top if it's more or less a routine treatment with a lot of experience already existing from other medicines. So risk assessment is always to be seen in the context of the particular medicine. And then we come to risk mitigation. So that obviously should ideally be a standardized or systematic approach within one development of a medicine, but also between manufacturers. So maybe there needs to be harmonization. A good example is for the recombinant clotting factors in hemophilia, where an immune response against clotting factors, obviously for patients with hemophilia, has severe consequences. So there have been even conferences with regulators and attempts to harmonize the approaches. Then the extent of the safety database pre-approval, how much do we actually know? Is it an orphan medicine, so a medicine against a rare disease where we cannot get so much data? And do we maybe have to take a consequence for the clinical trials? So for high-risk medicines with immunogenicity, do we have to power for safety rather than for efficacy, and so on and so forth? So that is more or less an individualized risk mitigation approach. And as I said, this is a living concept, so we have to revisit this during the life cycle. Now, as an example, here's two products, and this is on the y-axis. You see the incidence of an immune response. You can see this is way different between product 1, which has only maybe a percent of an incidence of unwanted immunogenicity, whereas product 2 is so immunogenic that you have in 85% of patients an anti-drug antibodies, ADA. So obviously there is a high risk and a low risk medicine here. The question is, which one is the high risk and which one is the low risk? If I was in a room with you now, I would ask you who is in favor of saying that product 1 is a high risk product, and there's usually a few people flagging up. And for product 2, some people would say, well, we don't know because we have to have more information. That is indeed the point. So it's not just the incidence of the immune response. In a risk-based approach, you need to, as I said, know the consequences as well. And indeed, as you could almost guess, product 1 is the high risk product, and product 2 is the low risk product. So product 1 is indeed erythropoietin, which I have already talked about. So here, even in a few patients only, if the risk and the consequence, if the consequences are severe, this would be a high risk medicine. So in this example here, it was only 2 patients in the entire safety population, which was 314 patients. So less than 1%, but still of critical consequence. Whereas the other product is a monoclonal antibody called alemtuzumab or Lemtrada, which is indicated in relapsing remitting multiple sclerosis. And interestingly, also it's a humanized monoclonal antibody. It was very immunogenic. Very likely one of the reasons for that was that patients are treated not constantly, but in treatment courses. So we're basically almost like a vaccine. You would give this in short courses or only a few times, and then you have a break in between. And this is an antibody which is against CD52, a cell surface antigen on T cells and B cells. And interestingly, the majority of patients, as you have seen from the graph, in the pooled phase 3 study analysis tested positive for anti-alemtuzumab antibodies, most of these actually with inhibitory antibodies. So what does this mean for the patients? Actually, interestingly, this is such a powerful B and T cell modulator or depleter that the immune response, if it's mounted again upon re-exposure, doesn't even have enough time to be mounted because the B's and T cells are already depleted. So when you look at the efficacy data, there was considerable efficacy in multiple sclerosis overall. There was, at least at the time of approval, no impact on safety in the database. No infusion reactions because there was methylprednisolone pretreatment. No impact on T cell depletion. What's the pharmacodynamic effect? No impact on magnetic resonance imaging, which is one of the readouts that you normally do for patients in multiple sclerosis, and no impact on efficacy, as I said, also on secondary endpoints. Part of assessment of this is that if this was a chronic treatment and it had such a high immune response, you could actually detect it with doing regular MRI scans. So that is part of the risk mitigation if that was a problem here. And there's also availability of alternative treatments. And very likely, as I said, the rapid depletion before any immune response can wake up is very likely the explanation of why this doesn't impact at all. So that's a good example, I think, for high incidence but lower risk. Now, risk-based regulation of medicines is not new. Sorry for showing you a picture like on the left-hand side. It is just a case which is quite long time ago now. So that's the TGN 1412 case. That was a monoclonal antibody against CD28. That is basically a polyclonal T cell activator. In this case, it had an unpredictable activation polyclonally of T cells, which resulted in cytokine release and a shock-like syndrome in patients where basically then the circulation was so bad in the patients that they had a necrosis of the fingertips and the toes. And we have an own lecture of with this for first in human clinical trials, the selection of the dose, sequential inclusion, and so on. So that doesn't mean that all antibodies or all immunoglobulins are high risk. So for intravenous immunoglobulins, which is a very long-standing blood-derived medicine, this is a classical low-risk medicinal product. So an antibody structure doesn't yet make it a high-risk product. It is rather the mechanism of action, but of course also the structure inherently and other factors. So that is not new. We have been regulating these medicines accordingly. Immunoglobulins have comparably low regulatory hurdles when being authorized as compared to new monoclonal antibodies with a new mechanism of action. Another example of a risk-based approach, I think, which is useful, is another medicine against multiple sclerosis. So this is, I'm not sure if there's any experts in multiple sclerosis in the audience. If so, please forgive me, because this is a very, very simplistic view on multiple sclerosis. Basically, it's got a lot of different mechanisms, B-cell immunology, T-cell immunology, but one of the factors is actually T-cells migrating into the brain after having been activated, for example, by autoantigens, or external antigens. Then they migrate into the brain across the blood-brain barrier, and then they start to attack the myelin around nerves and around neurons, better said, not nerves, neurons in the central nervous system, and they demyelinating it and causing the multiple symptoms that multiple sclerosis actually has. And the way they do this, these lymphocytes, is that they adhere to the endothelium in the blood vessels via molecules which are called integrins. And one of them is alpha-4 beta-1 integrin. And that helps them kind of dock, like stopping at the roadside in a parking slot, and then get out and take the exit into the brain. And natalizumab is a monoclonal antibody which has been authorized for quite some time now. That blocks basically this integrin. So the lymphocytes cannot enter the brain anymore. They're just circulating, and they cannot be pathogenic anymore. And in early publications, now actually 20 years ago, natalizumab showed, and it's still one of the most efficacious medicines, very, very high suppression of new lesion developments. On the right-hand side, you can see the placebo in this paper. That is a steady increase, so very active multiple sclerosis patients where there is new lesions coming up almost every month. But with 2 doses of natalizumab, they are almost completely suppressed. And on the bottom, you can see the lesion development. It's the white spots there in the white matter of the brain. In MRI, the white matter is actually gray, and the gray matter is actually white. So it's other way around. But you can see these very white spots are the inflammatory lesions. And in patients treated on the right-hand side, you can see there's much fewer lesions. On the left-hand side, you can see a normal brain as an as a comparison. And of course, we were very happy at the time when I was a regulator. This came for approval because we hadn't seen such a high efficacy in multiple sclerosis yet. But it was almost a nightmare for a regulator. During the assessment procedure at the time, 2 patients died of a very rare disease, actually, which is called progressive multifocal leukoencephalopathy, PML. This is a very rare progressive disease, which is caused by JC virus, John Cunningham virus. This is a virus where almost all of us are infected already as children. It's dormant. So as long as the immune system works, it's kept in check. But as soon as there is a severe immunosuppression, like HIV or AIDS, it's one of the AIDS-defining infections, actually, or transplant recipients or patients with chemotherapy, it can be activated again and then migrate into the brain, and then at several places in the brain, so multifocally, attack the white matter of the brain. And you can see the MRI for the patient on the bottom right. There's a massive lesion there, which of course is self-explanatory that this is a very dangerous and critical clinical condition, which was very often fatal. And we have increasingly seen this now with biologicals. So natalizumab, Tysabri, is the brand name, was not the first one, but the one which put this much more center stage. So we have a situation where we have a certain risk for a very severe clinical condition, but we have a very high benefit for the patient. And this is a very interesting and important factor as well when we talk about a risk-based approach. We have a new medicine in front of us, that we also have to see the benefit of that particular medicine. So not only what does the risk mean for the patient, but also what does the efficacy actually mean for the patient. And this was actually authorized in Europe then, despite the known risk to induce PML. The reason was self-evident, as from what I said, clinically very relevant efficacy. And actually a risk-based approach at that time was then applied also in a late stage of development. So the limitation to patients with high disease severity without alternatives was actually the most important risk mitigation. So the indication was then that Tysabri is indicated as single disease-modifying therapy in highly active relapsing remitting multiple sclerosis. And then for the following patient groups, so it was even further restricted. So patients with high disease activity despite the standard of care, better interferon, or patients with rapidly evolving severe relapsing multiple sclerosis. And it says here C section 5.1, that is from the summary of product characteristics in German, Fachinformation. There is just more information which defines more what actually highly active disease is. So in this case, what was done was that the authorization was restricted to those patients where the benefit risk estimation was especially positive. And actually, this is an old screenshot from the very old European Medicines Agency website. I have not put this into a new screenshot because it's not so prominent anymore on the website of EMA now. But it shows very well what was done at that time, because you can see that there is conditions imposed on member states for safe and effective use in all languages, almost all languages. So basically the doctors and the particular member states had a duty there. They needed to set in place the risk mitigation plan. And this is a very important aspect. So pharmacovigilance, that is the vigilance of medicines authorized, that is spontaneous supervision of medicines, but also certain training materials and measures that the doctors and the multiple sclerosis centers actually had to undertake. So in this case, the member states needed to ensure that there will be a national and prior to marketing physician pack, product information, but that's needless to say, physician information, but also patient alert card. So the patient would basically have a little document with them, which they would bring to the doctor in case there is a problem, so that the doctor knows, okay, this patient actually receives natalizumab. What is natalizumab? And then they can read what it actually is. So not every doctor would actually know what natalizumab is. And the interesting thing here, which I think helped in risk mitigation, was that the treated disease, multiple sclerosis, is targeting the same organ as the adverse event, PML. So basically, if a patient has a developing PML as an adverse event, they would go to the neurologist, whom they know, and the neurologist, of course, then can be alert. The tricky thing is that the symptoms of multiple sclerosis and also of PML can be pretty similar. So it's also the clinical cause, how acute is it? So it requires a bit of training, and this is why there is also more information in the training material for the particular multiple sclerosis centers, because it was restricted not to regular neurologists, but to treatment centers with special knowledge. That was one of the other risk mitigations here. And you can see that there is quite a bit of information that the physicians would actually get. So not just about PML, but also about all the other problems that can actually arise. And then, as I said, a risk-based approach is not done once. It is done regularly. So it was actually then after a few years, actually almost after 10 years, Another major regulatory review of Tysabri, because new knowledge has come up and also tests have come up, because obviously it would make sense if this is caused by a virus to test if patients actually are positive for the virus. It's not that everyone has it. It's many people have it, but not everyone. So basically, if a multiple sclerosis patient does not test positive for JC virus, they should actually have a much lower risk. And actually over the years, that was part of the risk assessment after authorization. More and more data came up to show that indeed it is as suspected. If you don't have JC virus, you have obviously a much lower, if any, risk for getting PML. It's not zero because patients can still contract the virus afterwards. So this is why it's not zero. And also what is almost evident and logical, the longer you treat with Tysabri, the higher the risk. So basically the duration of treatment, the testing for JC virus, but also any immunosuppressant before starting Tysabri, even adding more to immunosuppression, overall are risk factors. And this actually helped. I will not go through this table in detail, but basically what it says, the longer the treatment was, and there was a testing, the patients, we could actually calculate the individual risk, not the individual risk, but the likely risk for a patient. So that was basically between 1 in 1,000 and 0.1 in 1,000, but obviously much higher for those patients at risk. There's two interesting factors here, two interesting things. When you have a test, obviously it needs to be reliable. So it is good to have a standard for the assay. So those of you who work on assays and assay development, there is also international standards very often for such pathogens or for other factors. So that's always good to have a look. So the WHO standard for JC virus, for example, on the right-hand side. And the other factor is actually how the patients themselves perceive the risk. So there was a publication made quite soon after authorization of Tysabri, where patients and doctors and regulators, I think even, were interviewed for the acceptable risk. And interestingly, multiple sclerosis patients accepted up to 10 times higher risk for themselves. And that, I think, shouldn't be neglected, because the patients know that if they have very severe disease, it's fortunately, multiple sclerosis in many cases is not a severe disease, but in some few cases it is. And then the patients know that they will have to take a certain risk if they can get a very active treatment. Of course, the patient should be heard as well. So you can see there's a lot of factors. Then the longer you go into the development of a medicine that can be used for risk identification and mitigation. And actually, there's even more to this, because PML might actually be a treatable disease. So obviously, viral treatments are not easy to cure or not easy to treat. But there was an article then a couple of years later in the New England Journal of Medicine where an anti-PD-1 antibody, so an immunomodulator, an immunoactivator, actually pembrolizumab, actually showed efficacy, at least in a number of patients with PML. So that was an interesting and very welcomed paper, actually, for the neurological community. Now, in the last part of this presentation, let's have a look at gene therapies and cell-based medicinal products, because when the legislation was made in the European Union, regulators and legislators actually had already learned about risk-based approaches and thought it would be a good idea to make this more systematic into the legislation. And actually, the Committee for Advanced Therapies, CAT, at the European Medicines Agency at the time, then worked on a guideline on that. They were asked to work on a guideline. And I can tell you that this was one of the most difficult guidelines to write. And the reason is of course understandable to you now that you have listened to this presentation. Risk identification is not the only thing. It's also about risk assessment and risk mitigation, but it's so many different factors that actually contribute to risk. So how would you write a guideline when you have so many different products at hand? And basically, you can read this from the text. When you read the guideline, you can see how difficult it's actually been to write this. Here, this was defined as a strategy aiming to determine the extent of quality, so that's pharmaceutical quality, non-clinical, that is animal data or functional data, and clinical data to be included in the marketing authorization application to justify any deviation from technical requirements. What sounds very dry basically means there are certain regulatory principles, certain studies developers have to do already early in development. But if there is a higher risk, it might be that additional data are required. But of course, as well, if there is a lower risk in the risk identification and assessment, then certain studies may actually not at all be necessary. And I think what the regulators at the time said was also that this is not a risk-based approach to the product as a whole. And that was important because they wanted to avoid kind of a stamp high risk on an otherwise very promising medicine, especially early in development, where small companies have one asset they develop and they get a stamp high risk by the regulators. Who would invest into this? So that was actually not at all the intention. And this is why it was important to highlight that this is more like a guide towards safe development rather than anything else. And this is a whole list of different risk factors, and these are even only examples. I will not go through all of them, but if you look at the buzzwords here, tumor formation, for example, treatment failure, that's for all medicines, but tumor formation, especially for embryonic stem cells, for example, who actually have as their own job, the only job to divide, that would be a tumor already by definition. And of course, then they differentiate. And if they can't differentiate, or if you have too many embryonic stem cells in a medicine that's derived from embryonic stem cells, then of course there's an inherent risk for tumor formation. And there's a number of other things like gene therapies, for example, can transduce inadvertently the germline and then be transmitted from generation to generation. There's toxicity to all sorts of different attributes, even completely agnostic of the later clinical use. So, for example, unintended alteration of cells by cell culture, degradation or leaching of toxic compounds from structural components, and so on and so forth. So basically what you have to do is the anatomy of the product. You have to not just look at the product itself, but also the way it's made. So for example, if you have products of or excipients, or maybe even only helping agents, so to speak, in the manufacturing process of animal origin, they might well be contaminated with adventitious agents, which adds to the risk. And this is why, again, product-specific risk factors have to be identified. Even a first step comes first, which is the risks for clinical use. That is what, of course, is familiar to you already, because it's the patient-related factors, the disease-related factors, as opposed to the product-specific risk factors. So it's basically more or less the same than what I've said in the beginning for unwanted immunogenicity. Take a similar approach here. It's the factors, the risk factors from the product and the risk factors for the patient. And then we are with risk assessment, which is mapping the data that are already there, or theoretical concerns, theoretical issues that might come up against the risks, and conclude on these risk factors and the risk relationship. So make an assessment, in other words. And actually the guideline is quite helpful because it provides a number of examples. So it says, interestingly, in the guideline that it is recommended that applicants wishing to use the risk-based approach communicate their intention to the European Medicines Agency and the rapporteurs, that is those basically at the time of marketing authorization. And in my mind, the risk-based thinking should actually be a reflex for anyone. So when you're very early, still in the lab maybe, or you are thinking about developing a medicine, you shouldn't think, this is later at the time of marketing authorization. No, because you would already now generate the data. You would already now be able to generate data that shouldn't lack later on. And In this annex for the guideline, there's a number of examples. The first one is an AAV, so adenovirus associated vector, pretty standard vector system, non-integrating into the genome normally, at least not to a large extent, expressing, and that of course is a little bit of a funny way, fictionase for a fictional disease, administered intramuscularly for the treatment of Fe deficiency disease. And actually already from this, you can see a number of implicit factors here that help you in the risk assessment. AAV vectors, low level of integration, well-known platform. So basically, we already know quite a bit of risk, or of the lack of risk for that. That is very helpful, but it's immunogenic. So basically, all of us have already had adenoviruses. Some of the nasty colds we have in winter are caused by adenoviruses, and this is why we need to take an adenovirus associated vector, which is a more exotic type. Otherwise, we would basically neutralize, the patient would neutralize it with their pre-existing immune responses. But of course, they might cross-react. So there is a certain risk for loss of efficacy. And there is a low but existing risk for tumor formation and a low potential for insertional mutagenesis. So basically, that is from the vector platform, a relatively low risk product. The transgene is human, so it's human fictionase. That implies it's got a low risk for an unwanted immune response. This time, not against the vector, but against the transgene. So for gene therapies, you see you have two products. You've got the vector, to look at the vector, and you've got to look at the gene of interest, what of the protein that's expressed. But Fe deficiency disease, of course, is maybe a tricky problem here. So there's a patient-related factor here, and the patients are, as it's said, as the disease says, deficient for Fe, whatever Fe is. It's of course a fictional disease. So depending on the gene defect, their immune system may or may not be tolerant. And this is why sometimes in gene therapies, you see that in the indication wording, product XYZ is indicated for patients with residual residual enzyme activity. And that implies where the immune system is tolerant. Because otherwise, if there was a complete loss, if they didn't have the gene at all, the immune system would not be tolerant. And of course, there would be very high immune response against this, and it might not work at all, or it might get all sorts of different clinical problems. But if it's, for example, just a point mutation in the active center of the enzyme, which makes it completely inactive, but still the overall structure, the gross structure of the enzyme is still more or less the same as the natural compound, the immune system will be tolerant. So you see, there is certain factors, certain thinking required for a risk-based approach. But I would say this is a pretty low risk scenario. Let's look at another one, which is human embryonic stem cell derived cells secreting bioactive substances injected into the central nervous system. So human embryonic stem cells, as I said, they have a known inherent risk for teratoma. So this is what they do. As I said, the embryonic stem cells should divide so that they form an embryo. That's the physiological function, as we all know. And you can use them in order to differentiate into any type of tissue. And then they can also secrete bioactive substances. And they are obviously cultured in the presence of growth factors in feeder cells. And these growth factors might alter the cells as compared to the physiological embryonic stem cell. And also the feeder cells might do something. So there are increased risk for chromosomal aberrations to do cell culture, even especially if it's prolonged culture. That, of course, the longer culture is, that might reduce the risk for adventitious agents because you kind of dilute it over time, but still there is a certain risk. The growth factors, as I said, there's a certain risk again for this exposure. And basically, if all of these embryonic stem cells would then differentiate, then there's no risk anymore for teratoma formation because there is no embryonic stem cell left. But if not all of them differentiate, then obviously there might be still embryonic stem cells left as contaminants, you could say, and of course they injected into the human body can then lead to teratoma formation. And as I mentioned, the feeder cells are part of that as well. And then injection into the central nervous system obviously is almost a worst case scenario. So a clinically critical anatomical location, there's the blood-brain barrier that I've mentioned before, with the multiple sclerosis example is basically closing this anatomical compartment for the immune system normally. So if there is anything happening, it might go unnoticed more or less. And there's risk, obviously, for unwanted proliferation. The brain is, as we all know, in the skull. So if there is a tumor developing from embryonic stem cells, it might lead to increased brain pressure. So you see there is quite a number of different considerations which would rather lead to a higher risk perception and additional studies, how many embryonic stem cells are there, what chromosomal aberrations might be there after cell culture, and so on and so forth. So this requires quite a bit of differentiation, investigation. This, again, fictional medicinal product. And this example now, I think the last one is autologous chondrocytes in suspension for the treatment of articular defects due to trauma. So typically in the knee joint, sports people, but also less sporty people might have certain defects in the cartilage, which is not just painful, but which leads to quite some disability. And that can come up also after trauma, after a bone fracture, for example, which affects the knee joint as well. And the idea is here to take chondrocytes with a biopsy from healthy cartilage, to culture them and expand them, and then to put them back in this case as a suspension, not seated on a matrix. So let's again look at the risk for such a product. I should say that such products are authorized. So these are real-world examples, and from the fact that they're authorized, you can already see that the risk is medium to low for that, I should say. Autologous is very often associated with low risk. So many companies say, well, we've got an autologous product, kind of implying, well, we don't have any risk. But of course, that's not always the case. We have to really look at this. The immune system is tolerant. So if there's anything happening with autologous cells, the immune system will not attack them normally unless they have any neoantigens expressed or whatever. So that is a factor which is not relevant here, but just for any other autologous product, it's not a given that an autologous product inherently has a low risk, but usually they do. We do have historical experience with these products, or clinical experience. I mentioned in one of the very early slides in this presentation that available knowledge is an important mitigation factor. And cell culture and exposure to growth factors, of course, may again add to risk. So if you have, like in the previous example, a long culture of these cells, they might not just dedifferentiate, they might also have chromosomal aberrations and so on, which may or may not lead to problems. And the fact that these are in suspension, that's a bit of a simpler product without matrix. So we don't have to look at any matrix product that would have its own risks then. But of course, it may actually add to the risk, because a suspension to be injected into the knee joint might actually lead to the cells either just inadvertently being administered somewhere else, for example, getting access to the bloodstream and then migrate somewhere completely different, or they might actively migrate, which is called biomigration. Chondrocytes might actually not be prone to walk somewhere, so to speak, but other cells in other examples might well do. So that is a factor which has to be also looked at, and this is why for such a product, a biodistribution study, for example, in animals, might actually be helpful. And articular defects due to trauma, let's look at the indication and the patient-related factor. That's not a life-threatening condition. It's maybe chronically disabling, at least in more affected patients, but it's younger patients typically, and they have normal life expectancy. So you need to look at the risk assessment here pretty closely, as compared if it was end-stage cancer patients with no alternative. So you see, it's also the kind of wider clinical application that's necessary to look at. But with these examples, I think one could plot a quite nice argumentation for how the risk-based approach actually look like, what studies may be necessary or may actually not be necessary when engaging with the regulators and talk with them about the requirements for the particular product. So that's the end of the presentation. My take-home message for this presentations are: have risk-based thinking as a reflex. It's something which is always helpful because it helps understand the particular product or the particular procedure at hand. Do the anatomy of it. And always remember that risk identification is not the end. Oh my God, we have identified a risk. That's not where it ends. That's where it starts. And risk is either acute or chronic. Risk is always married to benefit and has to be assessed together with it. And it's a continuous process, also in early translation, as I mentioned, and requires frequent reassessment. And early high risk may not mean high risk forever. And mapping the risk may actually help being systematic and assist in controlling the risk. So that's the end of the presentation. Thanks for your attention. If you have any questions, I'd be more than happy to answer them.